Hello everyone, and welcome to Ant Biology webinar series presented to you by Translational Outcome Research Group, Department of Zoology, University of Calcutta. As you are already aware of the fact that in this series we not only focus on different perspectives of biology, but also how biology is interconnected with various other disciplines. So to our dear audience, you can post your questions and interact among yourselves in the comment. We will convey our question to our respected speaker at the end of the session. And the feedback link would be also shared at the end of the talk in the comment session. So please keep a note on that. And our mentor and convener, Professor N.R.A. Banerjee, has sent her best wishes and regards to our respected speaker. And I will take this opportunity to introduce our eminent speaker, Dr. Upasana Ray. A very warm welcome to you, ma'am, in our platform. Dr. Upasana Ray is the Senior Scientist, Deputy Head and Assistant Professor of Infectious Disease and Immunology Division, CSIR, Indian Institute of Chemical Biology, Kolkata, West Bengal. Her current research interests include virus assembly, entry, and host virus interaction of flaviviruses and also the novel coronavirus SARS-CoV-2. She did both her bachelor's and master's in zoology at the University of Delhi and her PhD in virology from the Indian Institute of Science, IISc, Bangalore. She is the recipient of various prestigious awards, the most recent being the ACRB Power Fellowship 2021, ILDC AMP Women Excellence Award by International Leadership Development Council 2020 are only a few among many. She is also selected for the Commonwealth Science Conference 2021, nominated by INSA, and elected full member of Royal Society of Biology. She has published in numerous prestigious journals along with many patents. She made immense contribution to research on the ongoing COVID pandemic with her works highlighted in Nature India, news articles covered by multiple news platforms like Times of India, the Hindustan Times, Statesman, and Anandabajar Potrika. Invited for panel discussion by Rajya Sabha TV on the topic India Fights Back Tackling Mutated Strains. We are extremely honored to have, ma'am, you in our platform, and the stage is all yours.
Thank you so much, and I'm honored being invited, and it's my pleasure to talk about vaccines and the work that really took off from laboratory, and it was an example from a work done which went from lab to industry. So, without taking much more time, I'll just start off. But before I go to explaining about the work, what I do is I'll give a brief background about vaccines. And since today's focus is virus-like particles, I'll then explain a bit what are these virus-like particles and how these candidates are developed, and then I'll come to the work. Okay. So, since there is a mixed audience and students are also there, this is just a brief overview about vaccines and types of vaccines and VLPs. So, we all know that what a vaccine is now, especially because of this COVID-19 pandemic. We have heard a lot about it. A vaccine is essentially a biological preparation that provides immunity to you, right? So, why we say biological preparation? We call it as a biological preparation because this preparation comes from biological components of the pathogen itself. Here, I'm talking in terms of the virus. So that's why biological preparation and the function. It has three major function. It stimulates essentially body's immune system, but it helps in recognition of the antigen as foreign. It destroys it for sure because antibodies are involved. And a very important component of any vaccine that you develop is to induce memory in your body. So memory is extremely important. Why it is important? So that your body can recognize the pathogen as a pathogen, and if you get challenged by this pathogen in future, then your immune system is going to remember it and get triggered and quickly elicit immune response. So a vaccine helps in recognition of pathogen as foreign, remember the pathogen as foreign and as a pathogen, and destroy it. Right. So now, what is or what are source or sources of vaccines that we develop? So here is an example of the pathogen. Here I have drawn virus here, but it can be bacteria or any other pathogen in that sense. So the vaccine, when we engineer it, can be engineered from various elements of the pathogen itself. The pathogen here, as I'm talking in terms of virus, is made up of proteinaceous shell, which is called as capsid. Sometimes some viruses do have a lipid envelope on top of it. So here in you can see a shell, icosahedral shell. So the structure is called as icosahedral. It's like a gem. Right. So this particular structure is a proteinaceous structure, but on top of it you might also have a lipid envelope, lipid bilayer, that the virus attains from the host cell itself while it comes out of the host cell. So that envelope has viral proteins embedded into it. So any of those proteins, or this or the proteins, protein or proteins which form the capsid or this proteinaceous shell of the virus, can be sources of various vaccine candidates, which is protein based, and inside the proteinaceous shell, which we term it as capsid or nucleocapsid, there is genetic material of the virus. So this genetic material can be DNA or RNA, double-stranded DNA, double-stranded RNA, single-stranded DNA, or single-stranded RNA whatsoever. So even the genetic material can be the source of vaccine candidates. So anything and everything that forms the part of the virus can potentially be vaccine candidates for engineering vaccines. Now, uh, so when I talk about protein, it can be the entire protein. Say, for example, this virus is made up of. We take the capsid structure, and the capsid is made up of protein A. Now, that protein A can be used as a the entire protein A itself, that's the whole protein, or you can protein is essentially made up of different domains. So you can also use a domain, one or two domains of that protein, so you're not using the entire protein in that sense. Or you can also use parts of the smaller, even smaller parts of the protein as a peptide. Now, nucleic acid can be used as a DNA form or in RNA form. Ultimately, what you aim for is a protein or a part of protein of the virus that this DNA or RNA will encode. Okay, so you know the central dogma that from DNA you form the RNA, and then from RNA you get the protein. So the end result is protein, but how you are delivering the vaccine, it can be either protein, it can be DNA or RNA. You already have heard about RNA vaccine in case of COVID, so you are already well versed of that RNA vaccines exist. So those are nucleic acid based vaccine candidates. Then you also have this pathogen infected cells. So there are different work published where people have used the entire pathogen infected cells as this as the source. Okay, so now, based upon what I said, the vaccines can be divided into different types. You can have inactivated vaccine, you can have live attenuated, then protein subunit vaccines, I just spoke about it, virus-like particles that we will be talking about today mainly. Then you have virus vectored vaccines, which where you use different You can use a different platform or a different virus where you inside this particle you have a DNA or RNA which encodes protein or protein that comes from the pathogen that we are targeting. Okay, so then you have mRNA or DNA vaccines. Inactivated vaccines, what are they? They are nothing but so inactivated vaccine and live attenuated vaccines come or fall under one bigger category of vaccines called as a whole organism vaccine. So as the terminology itself suggests, the whole organism vaccine would mean that you're using the entire pathogen as the source of your vaccine candidate. So here now, if you are talking about inactivated vaccine, that would mean that you are inactivating the virus. So that can be done by chemicals or radiations. So there are different methods. So here you are making this virus incapable of transduction or entry into the host cell, and also the genetic material is destroyed, so it cannot replicate. So it becomes non-infectious in nature, but it retains all the components of the virus. So that's inactivated. And then you have live attenuated vaccine. Live attenuated vaccine means that say you have a pathogen, say if I talk in terms of virus, you have a virus. Now you cannot use this entire virus in its pathogenic form as vaccine because you get infected otherwise. So what you are going to do is you take the virus and you force this virus to get adapted to a new host. If say I'm developing a vaccine against human, that I then I would use another host and I'll make this virus grow or multiply in the new host system. So slowly what happens, this virus attains some genetic changes and it adapts to its new environment. And when it has adapted enough, at that point of time we take out the virus from this new host and they put it back as a vaccine in humans. So when we do that, humans won't get the disease. So when I say human won't get the disease, that means that the virus can still transduce or infect or enter the host cells, but the replication is very slow, so much so that it cannot cause the disease. And since the replication is low, the immune system gets enough time to get primed. So that's live attenuated. Okay, so these are the different ways vaccines are engineered. Now, coming to today's topic, virus-like particles. So, as I was talking about different types of viruses, viruses can be of two major types: enveloped and non-enveloped viruses. I just told you in case of non-enveloped viruses, you have only a capsid. The capsid means if you see here this shell in yellow, so that's a protein. So proteinaceous a protein shell that is called as capsid, and inside is the genetic material. So a lot of times we also call it as nucleocapsid. So that's non-enveloped virus, and in case of enveloped virus, on top of this capsid, you also have a lipid shell, and in that lipid shell again you have different proteins of this virus that's embedded. It can be envelope protein, then you have matrix protein. So there are different types. Okay, so based upon these different categories of viruses, you can also have vaccine candidates which can be engineered with different permutations and combinations of all these components. You can have non-enveloped VLP where you are not using the envelope, and you can also have enveloped VLP where you are using the components of the envelope, or you are using the components of the envelope and also the capsid, but minus the genetic material. So since the genetic material is not there, virus-like particles look like viruses, but they are non-hazardous. They cannot cause disease. Okay, sometimes it is difficult to assemble this kind of the entire virus, um, using self-assembly system. So in that case, we can also make virus-like particles by using some other virus as the platform and just display the proteins from the pathogen against or from the virus against which we want to engineer the vaccine on this particular platform. So again, it looks like virus type, but it is not native virus. The beauty of this system lies in the high density repetitive display of antigens. So this kind of antigenic display on a platform, which can be the entire virus minus the genetic material, which is obviously just the virus, non-infectious form of the virus, or any other platform that can display the viral proteins, structural proteins in a repetitive high density manner. So now, so as I was saying, it's high density surface antigens is the crux of the entire thing. So when your vaccine candidate has can display the viral protein, uh, whole protein, or pieces of it, or whatsoever your antigen, to be very precise, in a high density repetitive manner, your immune system gets better triggered. So so you can see here in the diagram, you have a B cell here. So if you see here, this is a high density repetitive display. So what's written? Critical number of receptors engaged, off rate. Okay, then here at the bottom you see the display is more sparse. So in this case, if you see these B cell receptors, not too many of them can attach themselves on the antigens, in contrast to figure A. So that's the difference. Okay. Now, so your body, or our body, our immune system gets educated when it in early stages not to recognize self antigens as foreign. So we do not produce immune reaction against our own proteins or our own components. And when that happens, we if we elicit our immune system elicits immune response against our own proteins, then we get autoimmune disease. We all know that. So uh, many the earlier work on vaccines, there are some group and there are some very important papers that came up where researchers have shown that if you take your own protein against which you generally don't elicit immune response and display in high density repetitive manner, then you are able to cross the self tolerance, and with the antigen density, your body actually elicits immune response against your own protein as well. So what's important here is the display of the antigen in a high density manner. Okay, so why I emphasize so much on that is because that's something important that has been used as a concept in developing virus-like particle-based vaccines, and why they are so important. So I was lucky working in a group at National Institutes of Health, Bethesda, United States, where the deputy chief of the laboratory that time, Dr. John Schiller, is very well known for his work on human papilloma virus vaccine, and he is the pioneer who developed the concept of HPV. VLP vaccines, and then this technology was taken up, and this, the two vaccines that are now there in the market, Cervarix and Gardasil, came up. So that's the product of this laboratory, and I was fortunate enough to work in that laboratory, and my direct mentor that time was Dr. Christopher Buck. So we got the idea from this HPV vaccine. So that's how that lab started. flourishing in the vaccine field, VLP-based vaccine field. So after this HPV vaccines, still they are working on next generation HPV VLP vaccines. So the project that I will be discussing today, or the work that I will be discussing today, is what I started off there, and in 2019 it was finally licensed off to Biologic Limited. So my work is not about HPV vaccine, but the concept is similar. So here's the concept. So if you have, so these are the structural proteins of human papilloma virus. So L1 and L2 are the two structural proteins of human papilloma virus that form the capsid structure of this virus. Okay, so what they showed was when you express these proteins inside mammalian cells, these proteins self-assemble into virus-like structures. Okay, so what happens if you're using L1 and L2? These are the two proteins, structural proteins of HPV, human papilloma virus. Uh, so L1 proteins assemble into these pentameric structures. So 5 L1 come together, they form these structures, and then L2 also join up, and they then self-assemble into virus-like structure. You can here either use L1 only or L1 plus L2. What happens when you use only L1? You get a structure similar to the virus, but this structure cannot gain entry in host cell. When you use L2 plus L1, this will form a structure. Although it doesn't have a genetic material, it can gain entry into the host cell. But since it doesn't have the genetic material, here the DNA that I am showing is something is a modified version that I won't explain here. It's not required. But then in the essence of the entire thing is that this particular type of virus-like particle can enter the host cell and but cannot cause disease. So one of them cannot enter, one of them can enter, and they have different. We have different uses of these two different platforms. Okay, now what I did, I started working on a progressive multifocal leukoencephalopathy. This is a neurodegenerative brain disease caused by replication of JC polyoma virus, which looks like human papilloma virus, and it's the structural proteins of this particular virus behaves similar to human papilloma virus. And my job was to develop a vaccine candidate, a VLP based vaccine candidate against progressive multifocal leukoencephalopathy. So as I was saying, that is the virus that causes this brain disease belong to a family of viruses called as polyoma viruses, or the family is Polyoma Viridae, and these are double-stranded DNA viruses. And so when they replicate in brain, it causes demyelination, and the disease is fatal. There is no treatment available. This is particularly important in case of multiple sclerosis treatment, or whenever a person is undergoing an immunosuppressive drug treatment. Then in immunocompromised situation, since the immune surveillance of brain is less, the virus gets ample opportunity to amplify, and then it causes PML. So that's why it is also difficult to start immune suppressive drug treatments unless because when you are treating a person for something, but then you end up catching another disease. Right. Also, in case of people who want to undergo organ transplantation, then you give immune suppressive drugs. In those people also, there is a chance that these patients can succumb to PML. Now, so let's see what this virus is. The viral structure is, so this is a non-enveloped virus, so there is no envelope involved, and this is how the structure of the virus looks like. So a virus can have two different types of proteins, structural protein and non-structural protein. And as the terminology suggests, structural proteins are those proteins which form the visible structure of the virus, whatever you can see. But when a virus enters a host cell, the shell ruptures and the genetic material comes out, and the genetic material also leads to production of other types of viral proteins called as non-structural proteins. So these non-structural proteins won't form the structure of the virus, but they help in the replication process, translation process, and virus assembly inside the host cell. Okay, so one of those structural proteins here is VP1. This is the major capsid protein of this virus, and as the name suggests, this is the major capsid protein. So the entire capsid that you can see here is essentially VP1. And then there are two more minor capsid proteins which is not easily seen, but they are embedded in between, VP2 and VP3. And just like human papilloma virus, these minor capsid proteins are important for efficient transduction. The terminology transduction means entry of virus in the host cell. But the major capsid protein is sufficient for the virus to assemble and look like the virus. So this is the capsid, or the proteinaceous shell of the virus, and so the capsid is made up of VP1, major capsid protein. This VP1, just like HPV, it pentamerizes. So 5 VP1 subunits come together and form this kind of structure that I have encircled in red. And 72 of these pentamers come together and form the entire viral structure. Okay, so what we did, we had to assemble this virus without the genetic material. Okay, so what we did, so in that, for certain experimental purposes, you might include DNA component, but that DNA is not viral DNA. That is a reporter, which can be luciferase, or it can be GFP, fluorescent protein, anything like that. That is not important. What is important here for you to focus on are these three plasmid structures here. So these are mammalian expression plasmids. I hope you all know what a plasmid is. So what we did, we cloned the structural proteins of this virus in different mammalian expression plasmids. And one important thing here was the presence of SV40 origin of replications is in this particular plasmid. Why this is important is the system, or the mammalian cell that we used here, 293 TT cells that we used here for expression of these viral proteins, had a stability. These cells could continuously produce SV40 large T antigens. So these were stably transfected cells which could produce SV40 large T antigens. The only important thing that for you to note here is that SV40 large T antigen would has the capability of binding to SV40 origin of replication, and that helps in replication or multiplication of a plasmid or a DNA inside the cell. So why that is important? Because when we transfect such plasmids inside this kind of cell, the plasmid will multiply. So since the plasmid is multiplying, you get more and more amount of viral proteins that you want to produce make. Okay, so what happens inside this host cell? Now you know that these plasmids will multiply, and then this plasmid will lead to production of RNA and then protein. The end protein is VP1, VP2, VP3. So these viral proteins will be produced in huge amount inside this 293 TT cells, and then due to the property of the virus to self-assemble, the viral proteins to self-assemble, you get virus-like particles inside this host, this cell. And then there are different methods. You lyse the cells and you purify these VLPs. Now, if you are having a reporter plasmid inside the VLP, this is also virus-like particle, but to differentiate two different systems. We call it a pseudovirus, and this we use for neutralization assays and other kind of assays where we want to actually visualize or see whether the virus has gone inside the cell or not. So what will happen when this virus gets inside the cell? Then the reporter protein expresses inside, and that's the readout of viral transduction. Okay, and to do that, so this kind, the pseudovirus, if you want to make a pseudovirus which is a kind of VLP, you have to use all the three structural proteins. But if you are only developing a VLP which doesn't have to really enter the cell, then you do not need VP2 and VP3. VP1 is enough. And that was the form of the virus or VLP that we used as the vaccine candidate. And pseudovirus is something we used for in our assay system to check the neutralizing antibody titer, etc. So like that, using this platform, we could clone and we could assemble almost 23 different variants of this virus. So I'll just walk you through the entire process, how these VLPs are made. So you don't have to really concentrate on this particular plasmid, the reporter. Just see that, just focus on this part, that is the plasmid expressing the structural protein of the virus. Here it can be VP1. This is essentially VP1. It can be VP2 and VP3 as well, depending upon you're making a VLP or a pseudovirus. So anyway, so the what you do, the first step is transfection. I have written co-transfection just to point out that if you are transfecting two plasmids or three or four plasmids, then that's called as co-transfection. But it's essentially transfection of plasmid DNA inside the mammalian cell. Here we have used 293 Tichy cells. So the plasmid will enter inside the inside the cell, and it will multiply, because if you remember, I talked about SV40 large T antigen. SV40 large T will bind to SV40 origin of replication and help the plasmid to amplify inside the cells. And from this, then ultimately end product is the capsid proteins. Capsid proteins will self-assemble and they form virus-like particles. Now, what you do, you lyse this culture, cell culture cells, and you harvest the entire thing. You use different detergents to do to prepare the cell lysate. So this is a crude cell lysate. That means there are different components of the cellular debris. Cellular debris is there, and then you have these. Nucleic acid elements also tangling and floating here and there. So it's a dirty preparation. That's why crude. So you have to purify your VLPs. How you do that? There are different enzymes available that would chew off these the nucleic acids which are not component of the VLPs, and you have so you are basically making the VLP free, and then you use ammonium sulfate to precipitate them down. So the entire this is called as maturation of VLP. So the maturation process is done at 37 degrees Celsius, and we did it overnight maturation. And then you clarify. Clarification means after the entire maturation process, you spin down the preparation, and then ultimately you do a density gradient ultracentrifugation. So in this step, we used iodoxanol gradient. You can also use sucrose or cesium chloride based upon how you have standardized your system. And OptiPrep is just a name for, so it's same as iodoxanol. So we did a density gradient ultracentrifugation using these different gradients. So you take the cell lysate. So there is a tube that is available. It's a kind of plastic tube where you layer the iodoxanol gradients in this form, and then on top of the gradient you overlay your crude cell lysate which has been matured, and then you do centrifugation at 50,000 rpm 16 degrees for around 4 hours. So at the end, what you get is you're somewhere in the gradient, generally in the middle gradient, your virus preparation, VLP preparation settles. So now you take a needle and you pierce through from the broad bottom and you collect fractions, multiple fractions, and then you screen them. So I won't explain how that is done, but so this is how, so when you purify the VLP and you check it under transmission electron microscope, this is how it looks like. It looks just like the native virus. So we got our potential vaccine candidate, and we had to test it. So first we went ahead and tested it in mice. So there was an array of mice immunization studies that we did using our VLPs, and we found that one of the VLPs that we used could neutralize to almost 23 different JCV variants. Now, here you might already be knowing that there are prime dose and then you have booster dose. So we did two dosages, prime and the booster in mice, and we checked neutralizing antibody profile in at both the times, and we saw that if you give two doses of this vaccine, it works best. So that was the preclinical studies in mice, from where we got the hint that our vaccine candidate could elicit good titer of neutralizing antibodies. Now, just let me explain very brief what is neutralization. I am using this term again and again. Some of you might not be knowing. So when I say neutralizing antibodies, that means when you are injecting, you are immunizing anyone, and you get you what you expect is the body will produce antibodies, but those antibodies might or might not be neutralizing in nature. That means it when we say neutralizing antibody, and that's what we expect from a vaccine to elicit. So antibodies, these neutralizing antibodies should be able to bind to the pathogen and stop the pathogen from entering the host cell. So if the antibody pool that gets generated because of our vaccine candidate is of neutralizing type, then we can depend on our vaccine candidate. And our immunization studies revealed that the antibodies or IgGs that were produced in mice could neutralize the virus. So we went ahead for a human trial. So this was a single human trial where this lady was in comatose conditions, and so after getting all the permissions, we vaccinated her with our vaccine candidate, and this was done in collaboration with Dr. Paola Cinque and Dr. Roland Martin. So they are from different places. Dr. Cinque is from Italy, San Rafael Institute, and Dr. Martin is from University of Zurich. So so what we did, we immunized this patient, and then we followed up for almost more than a year. So this scan shows MRI scan to see the evolution of PML lesion. You can see here in red arrow is the point of time where we immunize this person. So in white, if you just focus on the white lesions, and particularly here in yellow, this is I won't explain all these different types of MRI scans, but if you see the yellow part here at the bottom, so that's PML lesion, and after vaccination you can see that the lesion goes off. So the vaccine was working, and there was a progressive rapid extension of lesion from in the white matter bilaterally before the immunization or the vaccination. However, when we vaccinated this lady after the vaccination, so you can see the date 2012, and here we have 2013. 2013, you see regression of lesion. Okay, So this work was then published in Science Translational Medicine, and we applied for patent. The patent was granted in 2018, and in 2019, just when there was outbreak of COVID, so it was then not declared as a pandemic yet. So in October, this was licensed to Biologic E Limited, and that's how it was. So finally, here I would like to stop by acknowledging people who were deeply involved in this project. Dr. Buck, Dr. John Schiller, I have already told about them. Um, so Dr. Paola Cinque and Roland Martin were our collaborators. These are my current lab members, my students who are involved in different types of other vaccine work and even basic biology work. And funding came from funding comes from CSIR. I work at the CSIR Institute, so definitely CSIR, and then SERB, Science and Engineering Research Board, DBT, Government of West Bengal, and I'm involved in a lot of outreach activities. So the International Science Academy, International Young Academy of Science, Royal Society of Biology, and OWSD are the platforms with whom I do those activities. And with that, I would like to end, and it will be my pleasure to take up any questions if you have.
Thank you, ma'am, for your wonderful talk. And ma'am, I think our audience have got whatever you have said. So we do not have any questions today. And it's been a pleasure, ma'am, to have you on our platform. And ma'am, would you like to share any tips for our like students like us, and also students who are from BSc and MSc, how they can proceed in their careers? Perhaps those who are wanting to be virologists in the next generation.
Um, if you want to become a virologist, is basically that depends on the subject you have taken up now. Zoology, I am myself a zoologist, so I did my bachelor's and master's in zoology from University of Delhi. So pretty much any area of biology, if you are doing, ultimately what you need is your PhD. And nowadays, it's essential that you taste a sense of different laboratories outside the country as well. There are plenty of opportunities within India. Also, nowadays we have fellowships available. You have early career fellowships from TBT Development Trust India Alliance. So if you want to do something here, that also you can do. So you essentially have to finish your PhD and then take up a specialization. This postdoctoral training is extremely important nowadays, where you experience different laboratories that work on something specific that you are interested in that field, because nowadays you go very specific. When you say virology, virology itself is a big area. So what in virology you want to do? So yeah, so you go for your postdoctoral training, and then it depends. You want to join the industry, you want to do go for academia. So if you want to go for academia, then you know that you have to apply for faculty positions in different research organizations or universities. Or if you want to go for industry, there is a different that's a different path altogether. But there are a lot of scope now in India to, if you want to become a virologist. We still have only handful of them in India, not a lot of virologists, but it's an exciting field to work.
Yes, ma'am. And ma'am, I have one question, though I think it's a very common question. Ma'am, in case of COVID-19, What do you think? What will be coming up?
Um, I like what specific you want to ask. I am not sure. So, are you talking about vaccines? Or are you talking about the disease itself? What exactly?
Ma'am, both. I think both.
Well, in COVID-19, we already have a lot of vaccines in the market. A lot of vaccines that especially Covaxin and Covishield is there, then you might have read already in the newspaper. Serum Institute has promised Covovax very soon. So that's Novavax vaccine that they are making in India. So that's the unit vaccine. You have virus-like particle-based vaccines, also different types of vaccines coming up. mRNA vaccine is already there. uh, we are sometimes worried about these mutations and all, but whatever vaccine we have, it is very difficult to escape from the protection that a vaccine provides all of a sudden. That process takes some time. So if you are getting vaccinated, that your vaccine is going to protect you at least partially. So get yourself vaccinated, and mutations. A virus keeps mutating. It's its nature to mutate. So that should not be a matter of concern always. But what's important is to do surveillance and to do monitoring, to record the mutations that happen and see if the new variants that come up are being neutralized by antibodies elicited by the vaccine candidates that are now there in the market, the vaccines. So that's a long-term study, but I think what is important for us is to do surveillance and is to get vaccinated as early as possible.
Okay, ma'am. I think we have another question from audience, and yes, Dr. Anupam Ghosh wants to know how long VLP-like vaccine will be remain effective for particular virus, or more particularly for specific strain.
Yeah, so as I was saying, any vaccine in that sense, if I okay, coming to the question directly, VLP. So as I was saying, VLPs look like viruses. So it depends upon how you have engineered the VLP, what kind of antigens you have used to develop the VLP. Have you used all the structural proteins or a one particular structural protein? Whatsoever, VLP is a strong platform. A VLP or an inactivated vaccine or or a live attenuated vaccine, something that looks more like virus and something that displays the viral antigen in a high density repetitive manner. So those we expect that it should remain effective for quite a bit of time. It depends on virus to virus also, because each virus has a tendency of mutating at a different rate. So so it cannot be generalized when that strain or that variant will come up, which will escape the immune response. It is difficult to say that, but in general, effect of a vaccine remains for a longer time, and at least there will be partial protection for a long.
Okay, ma'am. So that was all questions we have today. And thank you so much for being in our platform. We are really honored. Our mentor and convener, Professor Rena Banerjee, has sent her regards and best wishes to you. Ma'am, we have another question. Can we take it?
Sure.
Okay. So this question is from Bijoy Vishwas. And do the current available vaccine are enough to tackle the upcoming COVID surge or a rapidly mutating virus?
This I think I have already answered. That it is very difficult to say whether something is enough to tackle, but it is. I think it is we are in a situation where we should not be thinking about that, because the vaccine, the pandemic itself started very recently, only last year. So, to get a virus strain that will escape, that will be called as immune escape variant, which is 100% immune escape, is difficult. It takes long time. And so the currently available vaccines, we should trust them, and I think that they will at least be able to protect you partially. When I say again and again, I'm saying partial protection, meaning even if there is some immune escape mutant, you should not be, dying in hospital if you are vaccinated. So even if you are infected, the intensity of infection and the progression of infection will be much lower than if you don't get vaccinated.
Yes, I think Vijay has got his answer. Thank you so much, ma'am. Again, we're really honored. And thank you so
Thanks.
much. And to all our audience, thank you for staying with us. And do subscribe to our YouTube channel and follow our website and Facebook to get updated about all our upcoming lectures. And we will soon have our next week series where we will be having Dr. Pakoli Bhattacharya and Dr. Gauri Shankar Shah, Professor Gauri Shankar Shah in our next series. So till then, stay safe and bye.